All right, so today we're going to be going over section 3, so final lecture, lecture 6. So as you guys know, in lecture you guys went over chemotrypsin, a couple other enzymes, and these are proteases that cleave peptide bonds. So moving into it, so before we get into the chemotrypsin mechanism, we have to understand kind of the role of the enzymes, how they work. And so in the previous lecture, I really hammered this concept of the enzyme introducing an alternative pathway that has a lower activation energy rather than the natural uncatalyzed reaction here. And because there's that lower activation energy, it allows for more of those reactions to occur more frequently. That's because, again, the enzyme itself introduces these favorable interactions with the substrate that allow it to convert into this product, or the transition state, if you will. Then there's this concept of binding energy, which is the energy released due to the non-covalent interactions between the substrate and the active site of the enzyme. And many non-covalent interactions drive the substrate specificity. So again, the enzyme has an active site, and the substrate is going to fit into that mold specifically. And it's based on these non-covalent interactions like hydrophobic interactions, dipole-dipole, hydrogen bonding, that kind of stuff. And to calculate the binding energy, all it is the difference between the delta G double dagger catalyzed, which is this one right here, and then delta G double dagger uncatalyzed, which is this top one. And the difference, that's the binding energy that you're saving because of the enzyme itself. Hopefully that makes sense. And then there's this Serine equation. I would recommend that you guys memorize it in case they ever give you values of the A, which is the frequency factor. If they give you values of the gas constant, then you know that. But the temperature, and they give you the delta G double dagger, then you can calculate what the K is. So moving on to the next slide, there's revisiting kinetic collision theory. I think this is really important when it comes to understanding enzyme kinetics and how specifically it can help with speeding up the reaction. So chemical reactions can only happen if collision between molecules are effective. So in this diagram right here to the right, we see that two molecules of A will collide with two molecules of B in a certain orientation, and if they do so, it forms an activated complex, which then forms your product, which is the combined molecules AB and AB. Whereas an ineffective collision, meaning that the orientation is not a side-by-side here, instead it's just directly one behind another. If these two collide, it doesn't form the products, and instead of forming this chemical transformation to now form AB, it's just going to bounce away. It's almost like an elastic collision. I'm not saying elastic because I know that means something different in physics, but I'm just using it as a loose term here. And so molecules are randomly moving in a cell, and without enzymes, it would be nearly impossible to begin chemical reactions necessary for sustaining life, since it's such an orderly thing. We're taking all the disorder, in the cell, we're taking all the disorder, the molecules moving in different directions, and we're making life from it. And so it's very hard to conceptualize that random disorderliness forming order, to form those polymers, to form glycolysis, Krebs cycle, etc., beta oxidation, all that stuff. So it's a necessary prerequisite that we have enzymes. So there are four factors to consider in uncatalyzed biochemical reactions. There's the random movement of molecules in solution. There's the reactants are surrounded by water in solution, which forms a solvation shell around the molecule itself. Then reactants may need to be distorted to break the bond. So for example, when the A and the B's collide, they could bend in a certain direction that allows the chemical bonds to break apart. And then lastly, functional groups need to align at the appropriate distances and angles. Again, this kind of plays too, we can have an OH here and another OH pointing out to the left, and if they collide, they kind of combine together to form this polymer instead. Whereas here, the OH is facing upward and facing upward, so they're not colliding at all. It's just the backbone per se. I'm just making this up on the fly. But know that these four are addressed with the enzymes, these four factors. It's on the next slide. So right here, how enzymes help with collision theory is that the random movement of molecules in solution, substrates are held in place with the enzyme. So here, the substrate can move in any direction in solution, but if they fit into the active site of the enzyme, again, you have those hydrogen bonds, you have those dipole-dipole bonds, you have those London dispersion forces, all that stuff. It's going to allow for favorable interactions between the enzyme and the substrate. And then for the second one, reactants are surrounded by water in solution. So enzymes allow desolvation of the substrate, which just means that water forms a shell around the substrate. When we put it inside the active enzyme of the enzyme, it sheds the water shell around it, if that makes sense. That's what this is representing right here. And so as you can see, there's no more water in this active site. We get rid of it. Thirdly is reactants may need to be distorted to break bonds. Well, if we have this induced fit model of enzymes, we see here on this bottom diagram that if, let's say, the enzyme was just a straight line, once it binds into the enzyme specifically, the enzyme's going to essentially distort the shape of it because of these favorable interactions with these little green pill-shaped thingies, whatever they're called. So yeah, they're going to form favorable interactions, going to distort the molecule, allowing for that cleavage to be more it occurs more frequently, essentially. So that's what number 3 is. And then number 4 is the functional groups need to align at appropriate distances and angles. Again, same thing with this induced fit model. If, let's say, for example, we're trying to break a bond connecting the two, or we're trying to bring them closer here. Like, this is just an example. I know this one breaks apart, but let's say we have an OH here and another OH, and we're trying to bridge them together. Well, because of this induced fit, it would be pointing down here, so they're not really close. But with this induced fit model, we see that they become closer, and it could like combine together to like form a, I don't know, a closer V shape. I'm just making this up on the fly, but this is another important thing of how enzymes address this. And then talking about the induced fit model of enzymes. So essentially, the enzyme, the active site, may not resemble the exact shape of the substrate, and it's only when the substrate binds to the enzyme that it induces the fit. It means that it changes it upon substrate binding to the enzyme. So conformational changes of the enzyme due to the substrate binding in the active site, enzyme has higher affinity for the transition state. And this makes sense. Our substrate is going to want to turn into product. In that transition from substrate to product, it's going to go into this transition state, and that's what the enzyme is best stabling at. Remember, that's what our binding energy is, that delta GB. Okay, and then for mechanisms of catalysis, we have a couple of general mechanisms of enzyme catalysis, and it's really good to understand the arrow pushing. No, it's not going to be an orgo to rehashing. It's just, these are the basic ones that you should know in case they ever ask you, like, draw a basic, for example, draw a basic general base mechanism. And you would just draw like a random histidine. It deprotonates the water, it makes it into a good nucleophile, attacks the carbonyl, because we know the carbonyl is electrophilic, and it puts a negative charge on the oxygen atom, and then from there it condenses and all that. So anyways, so you should know these 5. Condensation, which just means that if you have 2 peptides and you're trying to combine them, that's a condensation reaction. So it's going from this top thing to the bottom. We have hydrolysis, which is just the reverse. If we introduce the water, it'll break the peptide apart. And then we have acid-base catalysis, which I'm sure you guys already know about acid-base catalysis. If not, you can look up more things. These are very general, crude examples of that. Next is covalent catalysis, which involves like essentially your substrate actually forming a covalent bond with the enzyme. So right here, as you can tell, this substrate is alone. It's not bound to the enzyme at all. It may be in the active site, but it hasn't done anything chemically. Then after some arrow pushing and whatnot, you see that the substrate now is covalently bound to this enzyme. It's not necessarily through non-covalent interactions, it is through a covalent bond itself. So you'll see that covalent catalysis, that chymotrypsin enzyme mechanism that you guys are going to learn, it is covalent catalysis. It is a subtype of that. And then the next thing is you have metal ion catalysis, which just means that a metal ion helps to stabilize the substrate or the transition state in some way. So you use a free metal ion that's within the enzyme active site. And again, some of the goals of enzyme catalysis are it aligns the substrate into precise position and angles. The amino acid side chains provide chemical reactivity with the substrate. These amino acid side chains are found in the enzyme itself, and so that's what's going to interact with the substrate and can form these transient covalent bonds, again, like in this enzyme or in this covalent catalysis here. Next thing we have is condensation and hydrolysis. So again, as I mentioned before, condensation is just the forward direction of condensing these 2 peptide fragments into one polypeptide, whereas hydrolysis is just breaking apart the polypeptide into multiple fragments. And yeah, there's no like cofactors. It involves using water and OH. You can find a mechanism online about like condensation and hydrolysis just to refresh what the arrow pushing looks like, the arrow pushing diagrams, but that's about it for this slide. Then the next one is acid-base catalysis. So use specific acid-base catalyst, which it utilizes either protons or hydroxide ions from solution, and it's affected by pH, because obviously if you have hydroxides and you lower the pH, well, there's going to be no hydroxides any longer. And then we also have the general acid-base catalysis, which protons are generated by side chains in the transition state. So that just means like the histidine or like for example, lysine, arginine, you're going to have free protons that can be used in catalysis, and they'll be regenerated towards the end of the reaction as well, rather than grabbing the protons from the solution itself. Next is that covalent catalysis. I touched on it briefly, but basically the substrate becomes covalently linked to the enzyme as an intermediate, meaning that this is not a transition state right here. This is an intermediate. If we were to bottle this up, we would be able to like find this in a bottle, or we'd be able to put this under X-ray crystallography. We would not be able to put this middle species into X-ray crystallography because that negative charge on the carbonyl is going to want to condense, right? So it's going to almost spontaneously go kick back in and then break one of the bonds. If it breaks that enzyme carbonyl bond right here, this between the C and the X, well then it just goes back to the reactants, and we expect that. But if it breaks off the Y, then now we have this cleavage, and now we're forming this stable intermediate, right? And again, this is only the first half of the reaction. We still have to release now the substrate, or this product per se. We have to release this from the enzyme itself using water. So you utilize nucleophilicity from the amino acid side chain. So in this case, this could be a cysteine. It could be an SH, and when the H is deprotonated, it allows for it to attack as a effective nucleophile, right? So that's just an example. And then, yeah, you form this tetrahedral intermediate right here. Or, sorry. This one right here. It should be tetrahedral. Yeah, okay. It's... The formal nomenclature is called tetrahedral intermediate, but it resolves quickly and it can't be preserved because of that instability of the negative charge. That's the only reason why. So I know I've been saying it's technically a transition state, which you could think of it analogously, but the proper nomenclature is this tetrahedral intermediate. And then for metal ion catalysis, as you can see here, this is a better diagram, and an example of it is that the zinc is going to coordinate with that substrate that you have, and typically it just has to do with either ion interactions or hydrogen bonding, and it'll stabilize the substrate for better enzyme catalytic efficiency from the enzyme itself. So now moving on to some of the mechanisms that you guys should know. So the first one is Aspartic protease, which basically cleaves peptide bonds between two aromatic residues. So for example, HIV protease is a type of aspartic protease. And there are two asparagine residues in the active site that help with this cleavage of two tryptophan, or two aromatic residues. And so the way the mechanism works is that you're going to have your substrate here, right? So basically in this active site, you're going to have your substrate here, right? We have an aromatic molecule number 1. Although I don't know why proline is here, it's not necessarily aromatic, so it's probably not the best amino acid choice to do it with, but anyways, we're going to ignore that. So aspartate 25 is going to deprotonate a free water molecule to form a hydroxyl group. So we have this aspartate 25 here. It's negatively charged, remember, at physiological pH. So what it's going to do is it's going to grab this proton, and what that does to this proton's from the water will become a better nucleophile, and it'll attack the carbonyl, right? In the peptide bond. When the hydroxyl group attacks, it's going to basically kick the negative charge electron density onto this carbonyl oxygen. You're going to form that tetrahedral intermediate. Now, the really important thing to note is that the second aspartate 25 is going to form these hydrogen bonds, right here. And that's going to stabilize the negative charge, allowing that activation energy to lower down, essentially. So what this negative charge is going to do from the carbonyl oxygen is it's going to kick back. And yes, it could break this OH bond, right? But then you would just go back to this to the reactants, right? But in this instance, we're breaking that peptide bond now, where that peptide bond is going to grab onto the proton on this aspartate 25. Essentially what this is doing is it's resetting the aspartate 25 to be at the start right here. As you can tell from the products right here, that now it's negatively charged rather than still sustaining that proton. And again, that's where you see the cleavage. So basically you have a polypeptide right here in the substrates. Well, we want to cleave that peptide bond. So it's using this mechanism of a water as a nucleophile, and they're making the water a better nucleophile by using this enzyme itself and the aspartate residues to deprotonate it, and then it forms this tetrahedral intermediate, and then it collapses to finally break the peptide bond. So pretty much it. Aspartic proteases. Next one is serine proteases. So it cleaves peptide bonds again. All these proteases cleave peptide bonds, just a little bit different. So there are different types of serine proteases. There's trypsin, which cuts the carbonyl side after basic residues. There's elastase, which cuts carbonyl sides after small neutral residues. And then there's chemotrypsin, which we are going to go much more heavily into, which cuts the carbonyl side of aromatic residues. Again, this is important that you guys know and memorize what the basic residues are, the small neutral residues, and the aromatic residues, and then which types of proteases are going to cleave what. You definitely should have that memorized, because they may just say, oh, this is a elastase, this is a polypeptide, where is it going to cleave? And so they're not going to necessarily give you, cuts carbonyl side, small neutral residues. They're not going to give it to you. So some common features of all serine proteases is that common residues used to stabilize substrates. So a common feature of all serine proteases is that they use common residues to stabilize the structures and the substrates. and that's called the catalytic triad: histidine 57, aspartic 102, and serine 195. So you're going to see these 3 conserved amino acids within the trypsin, the chemotrypsin, and the elastase, which allow it to, again, it's essentially called the catalytic triad. It's these 3 amino acids that really do help facilitate the peptide bond cleavage. So now we're moving on to the chemotrypsin mechanism. I'm going to walk you guys through it on how it works. But first, before I go into like the nitty-gritty details, I want you guys to focus on the bigger picture, right? Remember, chemotrypsin cleaves a polypeptide into 2 peptide fragments at the peptide bond. So in this case, we need to find the peptide bond here, right? And it's going to be this carbon and this nitrogen. So this is our peptide bond that we're going to cleave at the very end. Now, like I mentioned, chemotrypsin utilizes this covalent catalysis, meaning that our polypeptide right here, pretend the Y was a nitrogen, right? Our polypeptide is going to be bound to the chemotrypsin, and it's going to stay bound at the intermediate. And this is only the first half, remember. So the second half is just going to be us cleaving this bond next. So keep that in mind. Chemotrypsin is like a two-step reaction. You have the first half, which is just to break that peptide bond, and the second half is just to release it from the enzyme. So with that in mind, we can start going into the actual specifics. So step 1 is that we always need the substrate to bind. It's not just going to magically appear there and then it's done. So the substrate first has to bind, and it does so because of this hydrophobic pocket right here. And it's in the active site that positions the substrates for non-covalent interactions. So this is why, again, it's a hydrophobic pocket. It doesn't want anything that's polar or charged or anything of the like. So the hydrophobic pocket has specificity for phenylalanine, tyrosine, tryptophan, and leucine. Leucine's the specific outlier, so you definitely memorize that. And again, as you can see here in this diagram, this is our phenylalanine right here. So it's gonna basically orientate itself, the molecule, to fit in this hydrophobic pocket. And so as you can see here, it binds into it, and it allows for the rest of the polypeptide to position itself properly within the active enzyme. And so that's what step 1 is. It's just getting it in this active site and then properly orientating it, so that way there's favorable interactions and the enzyme can do its thing. So that's the first one. So then moving on to the second step, there's the tetrahedral formation, where we're going to form this ester bond. Ignore all this like nomenclature stuff. Remember, what we're doing, keeping it in simple terms, is now we're doing these arrow pushings right here. This is where we're at now with the chemotrypsin mechanism. So right here, if we look at this diagram, we see that the way that the enzyme adds itself to the substrate to form the tetrahedral intermediate with that covalent catalysis, the way it does that is that we have the histidine deprotonate our serine. Our serine is the nucleophile in this case. And there's a difference. In aspartic proteases, as we saw, its own it basically does a similar effect, right? It will deprotonate the water becomes a strong nucleophile. But in this case, we're not using water for the serine protease, we're using the serine itself. So in this case, we see that the histidine deprotonates the serine. The serine is going to attack this carbonyl carbon next to the peptide, within the peptide bond, adjacent to it, and it's going to form this tetrahedral intermediate, and it pushes back these electrons from the carbonyl oxygen to form that negative charge. Again, that glycine 193 and the serine 195 form the stable hydrogen bond to stabilize this negative charge on the carbonyl carbon. If we didn't have these 2 residues, it would be very hard, extremely difficult for the intermediate to even occur and exist. So again, this is a short-lived intermediate. It's called acylation. So if we ever use that term, that's what it means. So again, I'll repeat this one last time. So we have histidine 57 deprotonate our serine, and the serine is going to become a strong nucleophile, serine 195. And what it's going to do is it's going to attack the carbonyl carbon that's adjacent to the peptide bond right here. And what's going to end up happening is you're going to have the double bond to that carbonyl oxygen. It's going to turn into a single bond, and the negative charge ends up on that oxygen. So this is what we end up with. So again, don't worry about these arrows that are denoting here. We'll talk about that in a little bit. But there's a negative charge here. We now have our polypeptide bound to the enzyme now. Remember, so this is another thing. It's covalently bound, not through these hydrostatic interactions. And this part right here is called the oxyanion hole, which stabilizes the negative charge on the tetrahedral intermediate, and it's necessary for this intermediate to be stable enough to happen at a faster rate. So this is all in words what I was just saying, but hopefully the arrow pushing diagrams now make a little bit more sense. So moving on to step 3, now we need to collapse that tetrahedral intermediate. So step 3, if we were to go back to this, we're right here now. We need to collapse it. So we have this negative charge built up. It's going to want to collapse and kick off. And again, remember, this Y is our nitrogen. So it's going to kick off that bond. So in this instance right here, we see that the negative charge kicks back, and it's going to cleave that bond between the amine side of the peptide, and it's going to basically cleave it off. And what's going to end up happening is that peptide bond is going to grab that proton from the histidine to reset it. Remember, this is very similar to, again, what we said about the Aspartic protease. When it kicks back that tetrahedral intermediate, that peptide bond is going to grab the proton from the amino acid residues of the enzyme, and it's going to reset the enzyme back to its natural state. So this is exactly what we're doing in this case, but when instead of doing aspartate, we're doing it for serine. So right here, step 2. So again, that peptide bond, or sorry, for histidine, not serine. So it's gonna grab onto the proton and reset the histidine. And then now the product, the first one, that amine side of the peptide, is gonna be able to dissociate freely. And then, so now that we have this like acyl enzyme intermediate, and it's stable here as it is. And the reason why is because we have the hydrophobic pocket keeping the peptide fragment bound. We also have this oxyanion hole being stabilized by the hydrogen bonds of the serine 195, but we also have that it's covalently bound as well. So this is something to keep in mind as now we're forming this stable intermediate. Terminal peptide, yeah. Okay. Oh, and something to keep in mind as well that I don't want to confuse you with is that the N-terminal peptide is still covalently bound to the serine enzyme. And remember what we said, it cuts the carbonyl side of an aromatic residue, right? So if, for example, we have this aromatic residue, we're saying that the amine side is on the left, right here, the amino group is on this side, and the carboxyl group is to the right. That's what we're assuming. So when we're talking about cleaving the bond, it's usually here, not over here. So something to keep in mind as well when we're talking about where it cuts on the specific side, whether we say it's carbonyl, which is this side, or whether it's the amino group, or our amino terminal right here. Okay, and then in terms of, now we're moving on to the second half of the reaction. So we have this enzyme acyl intermediate, but we want to release our other peptide fragment that's covalently bound. That's where we introduce the water again. So the water is going to basically break this bond. So that's the whole end goal, is that we're going to go from this acyl enzyme intermediate to now the fully released product right here, which it's no longer covalently bound. So moving into this, so the water is going to go into the active site, right? And it's going to be stabilized by that histidine again. But remember, because the histidine does a phenomenal job of stabilizing, or basically grabbing that proton from the water, it's going to make water an extremely strong nucleophile, or stronger than it is in its natural state. And so the water will attack that carbonyl carbon again. And again, it's going to form that tetrahedral intermediate with the negative charge on the carbonyl oxygen. And it's going to be stabilized by the oxyanion hole, same residues, glycine 193, serine 195. And what's going to end up happening is the water is bound now. It's that OH that's bound to it right here. Sorry, it's weirdly configured here, but right here, this OH is from that water, that free water that we introduced into the enzyme. So it's going to basically, that negative charge is going to kick back in, and then from here, instead of breaking the water bond, because if it does, then we just go back to the free water molecule, right? No, what's going to end up happening is that bond between the peptide fragment and the amino acid residue of the chemotrypsin, it's going to break that bond instead. And what's going to end up happening is that bond grabs the proton from the histidine again to reset it. And the serine will also be reset because we know that it was protonated originally. So that's what this next step is. I was kind of jumping the gun a little bit, is that the tetrahedral intermediate collapses, and then you're going to see that it's going to form this free peptide fragment, and it can now dissociate from the enzyme itself. And then now we have our fully reset chemotrypsin now. Our histidine is deprotonated as we expect it to be at physiological pH. The pK is like 6.2, physiological pH is 7, so it's going to be higher as it is. And then we also have our serine that's protonated as well, and it's coordinated with the histidine. And the glycine, it doesn't deprotonate or protonate or whatnot. So that's the reaction, the mechanism of chemotrypsin. Hopefully that makes sense as to how it works, but I also written stuff in words here as well. So just some key takeaways from the chemotrypsin mechanism. There's 2 stages. You have the cleavage of the peptide and release of product 1, which is the carboxyl side of the peptide fragment. The second stage is release of the peptide from the active site, and you regenerate your enzyme. So that's steps 4 through 7 on the diagram. So I have that listed if you want to go through it again and replay it. So some star players are the hydrophobic pocket, which allows for specificity of which amino acids that are able to bind into the chemotrypsin active site. You have histidine 57, which deprotonates the serine. and the water so that they can function as nucleophiles. And then we have the serine 185, allowing that nucleophilic attack of the polypeptide chain in the first stage right here. And then some basic recipe steps common to both stages. The histidine deprotonates something with an OH group and forms a nucleophile. So maybe they may throw another question of like threonine, right? It still has an OH group, so it could be similar, but the reason that threonine isn't as effective as serine is because it has that methyl group adjacent to that same carbon. So it's just something like that could be proposed as an exam question. Nucleophile attacks carbonyl and forms tetrahedral intermediate stabilized by glycine and serine. Intermediate collapses and the product releases. And then that's pretty much it for like basic recipe steps. And then for the supporting players, we have aspartate 102, which enhances that histidine 57 base function. And I know it's not shown here, it's kind of off the diagram, but it is shown here in the free enzyme. It allows for the histidine because it can flip basically like, so either like, for example, this right here, this is the protonated nitrogen right here. And it's, let's just say it represents my thumb. It could either flip like this or like this. But the reason why it stays like this conformation, the other nitrogen is pointing downward, is because of this aspartate right here. So that's another key supporting player that allows for the enzyme catalysis to occur much effectively, and that therefore concludes the catalytic triad. Now, glycine 193 is not part of the catalytic triad, but it does help stabilize the oxyanion hole for chymotrypsin. The catalytic triad talks about the serine proteases in general, whereas in chymotrypsin we have these extra amino acids that play an extra supporting role that's not found in other serine proteases. And then moving on from the chymotrypsin stuff, we're going to talk about enzyme cascades and signal amplification. So we see that for enzyme cascades, what we're talking about is that a certain signal will come in, like a GPCR, whatnot, and it's going to essentially, or this is a better diagram to elucidate that. So let's say we get a signal from a GPCR. We have one, this is a effector protein essentially. It's going to do something to a secondary effector protein, and it's going to basically amplify the signal. That's all signal amplification is. It's like one molecule can activate a downstream effect of like thousands or millions. And so it's called the multiplying effect, where you activate one enzyme, and then enzyme 1 activates basically 3 of them, and then each of those enzyme 2s are going to activate more and more and more. So you see this multiplication of it. Yeah, that's good. And then in terms of allosteric feedback and regulation, so at a certain point, certain reactions have to stop, and there has to be certain built-in mechanisms within the pathway. So that's what allosteric feedback is in regulation. So for example, if we're talking about this complex diagram, just look at the aspartate, right? You don't have to memorize all this, but just look at the aspartate. We know that it's going to have some effect on this factor. I don't even know what it is. See, it tells you that you don't need to know it, but it's going to have these downstream effects. Let's just say that we have, the end result is that we generate a lot of isoleucine, right? Well, let's say that we have too much isoleucine, because of the consistent aspartate signaling, right? Well, we wouldn't want to just continue generating isoleucine. So what it's going to do is it's going to go back to the enzymes that are helping transduce the signal from aspartate to this and inhibit it. So that way, even though the signal may persist, the effects are mitigated. And so the end result is that you decrease production of isoleucine. This is just an example. And then this is exactly how we can control enzymes that's involved in like glycolysis, for example. So it maintains an equilibrium, which it allows for a relative amount of product and substrate rather than one to dominate over the other. There's genetics, so like genes code for enzyme proteins, which allow us to regulate enzymes specifically. So for example, if the lac operon is the best example I can think of right now, if you would only express the lac operon if you have lactose present, right? That's pretty much like the basis of it. And so that can control already the enzyme concentration, its ability to catalyze the reactions. So if you have no lactose present, you don't even transcribe the lactose gene itself. Whereas if you have a lot of lactose, you want a lot of lactase enzymes to be produced, so you can conduct that reaction and lower the substrate concentration accordingly. Then there's the zymogen proteolytic cleavage, which just means like if we have a huge polypeptide that is supposed to turn into a protein, but there's this portion of it that we do not care about, and that's actually what's preventing it from folding into that stable protein conformation. We need to cleave that little portion, and that's what a zymogen is it's that full length containing the site that inactivates it. But the moment we cleave it, that portion out, we take it out, then the enzyme can fold in it on itself, like easily. So usually the cleavage is done by an activating protease, so that's something to keep in mind. Then there's reversible covalent modification. Think of like phosphatases protein kinases, phosphorylation, glycosylation. You're adding and removing specific groups via covalent bonds. So right here, it's in this diagram, these are covalent bonds, to an enzyme which can either activate or deactivate them. And the effect is enzyme unique and very specific. So we'll see cases in gluconeogenesis right here, or glycogenolysis. I think this is an example of glycogenolysis, where phosphorylase breaks down glycogen, and it has a kinase and a phosphatase that allow it to either be activated or allow it to be inhibited. So again, and then there's obviously allosteric regulation, which it's very analogous to competitive inhibition, uncompetitive inhibition, non-regular inhibition, or non-competitive inhibition. And then just a little bit more on allosteric regulation. Again, there's these two types of things called homotropic regulation and heterotropic regulation. Homotropic just means that the modulating molecule is also the substrate. So for example, right here, if for M, right, for example, our M is a positive modulator, and let's just say it binds onto, so homotropic modulating molecule is also the substrate. So essentially, the thing that allows those allosteric effects, right, that will also be the same molecule that acts as the substrate. So if you, again, this comes back to like maybe a lactose. I don't know if this is like a true example or not, but I'm making this up. But essentially, lactose is going to be the substrate. But let's say that if there's a lot of lactose, we want it to allosterically positively regulate or promote the activity of that lactase enzyme. So even though it's a substrate, it'll also bind to the allosteric site of that enzyme, and it'll speed up the reaction rate. So something to know. And then heterotropic just means that the modulating molecule is not the substrate. So like the best example I can give is this one, where isoleucine is not the same as the aspartate affecting enzyme number 1, right? So enzyme number 1, the substrate is the aspartate, the allosteric modulator is isoleucine. Those are completely different. Whereas if, let's say, for example, aspartate was also a negative inhibitor to this, then that would be a homotropic regulator. And then yeah, the allosteric regulation can be either positive or negative. They'll usually tell you, they'll be explicit about it, or it'll either be explicit within glycolysis or whatnot, or they will just tell you ahead of time in the question set itself. And then enzymes undergo conformational changes after a modulator binds. So again, think of like connecting structure to the function. If the structure changes slightly, and when we know that it's a positive modulator, well, the structure is probably conferring more to the transition state. It allows for that enzyme catalytic efficiency to be greater. Whereas if it's a negative, it may block the active site. It may change the conformation so that the active site resembles nothing like the transition state of the reaction you're catalyzing. I know it's a lot of words, but just know that the positive and negative regulators do induce this conformational change, which is supposed to bridge that structure and function of the enzyme itself. And then looking at a specific example, we're looking at ATCase and allosteric regulation. So from left to right, we're going from this inactive T state hemoglobin to the active R state hemoglobin. And so right here we have CTP, which is just a random molecule. And what this CTP does is that when it's bound, it inactivates the hemoglobin, right? But if we remove the CTP, we see that it's activating the hemoglobin into the R state. So again, CTP is a negative modulator, because if it's bound, it promotes its inactive state, whereas if it's released, it's the active R state. And then ETCA or ATCase has two modulators that produce opposite effects, right? Oh no, this is ATCase, sorry. I keep saying hemoglobin because I see an active T state, active R state, but it goes from the inactive to the active. So ATCase has two modulators that produce opposite effects. Please memorize this diagram. I was asked for my final, I don't know why, because this is exam 1 stuff, and for the final, like, it's not cumulative. And they asked it, and I had to have this diagram memorized. And so ATP is an ATCase allosteric activator. So before I even talk about this stuff, just look at the diagram. This should look pretty familiar. We have V0, so the initial reaction velocity, and then we have our substrate concentration. What does that look like? A Michaelis-Menten plot. So even though it's like sigmoidal, let's say, right? It's not necessarily hyperbolic. We're going to talk about these different conditions. So the middle one's going to be the substrate alone. We see that ATP is an allosteric activator, so it increases the binding affinity. As you can tell, like if we were to draw a Km, like let's say it's arbitrarily here, it hits this line first, meaning that the Km is lower. It's inversely proportional to binding affinity, so the binding affinity is higher. And so that's something to know, is that ATP is an activator. And then CTP is an ATCase allosteric inhibitor. So adding CTP reduces enzyme activity. Again, this is what this is. This is ATCase, not hemoglobin. That's my bad. When we have the CTP, it forms this inactive T state, and so it's an inhibitor. And you can see that the Km now, if we were to draw it right here, it's all the way out here, which the Km is much greater, which means that the binding affinity is much lower because it's inversely proportional. And then note that CTP contains a pyrimidine, whereas ATP contains a purine. So go ahead, review exactly adenosine, cytosine, thymine, and guanine, and figure out if they're a pyrimidine, purine, and that'll help you a lot, because if they say like, oh, what's the predicted effect of a GTP, right? Well, GTP is a purine, like adenine or adenosine, and so you're going to expect it to activate it as well. So it's something to keep in mind. So more CTP indicates higher levels of pyrimidine, and yeah, here's another side note as well. More CTP indicates a higher level of pyrimidine, which would be the reason to decrease pyrimidine biosynthesis, right? If ATCase is what creates our nucleobases, right? And I think I'm starting to remember this now. ATC and CTCase, all that, if that is creating your nucleobases, right? Then if we have too much CTP, then it's going to act as an inhibitor, because we don't need any more. We can control it. We can have homeostasis. We don't need to produce more of it. So that's what this is trying to hint at, the second part. But yeah, hopefully that's lecture 6. I know I went by really fast. I'm super busy today, but hopefully that gave you guys a rundown of everything at least. And then also, I'm not going to go through this, but I put in a bunch of high yield topics that you guys should go over. Just make sure, like, honestly, if you want to use this as like a rapid review, just be like, okay, do I know the amino acid names, abbreviations, properties? Go through each little point. If you know it, then you're good. You don't need to study it again. If you don't, that's something to look at within the other slides I posted earlier, if their early lectures are within this slide. And then there's also some practice questions as well that I posted here. So hopefully that really helped you guys out a lot. And yeah, I'm looking forward to office hours on Wednesday that we're gonna have so much TA resources. So please, like, bring all your questions. There is a lot of things that we can clarify. There's probably some things I've said that is wrong in some of these conference sections, just because I yap. But just go to TA office hours, ask on Ed. We're gonna be, monitoring everything, providing as much resources to you guys as you can. So good luck with studying, and I hope everything goes well.